All right, well, to extend that particular experiment, the Corona lab, along with his collaborators, came up with a more distinct method of synthesizing small pieces of RNA. So those pieces of RNA were 2 to 4 nucleotides long, and they linked them together using something called copolymers. Don't worry, an enzyme to make copolymers. Don't worry about the name of the enzyme. Don't worry about that part. What you just need to know is that you had these pieces of RNA. And they basically did a similar experiment where they were able to identify each codon that associated with a given amino acid. So UC, UCU, CUC, sorry. So here, these both have the UC. Here, it's in the beginning for serine, and then UC for leucine, UC for arginine and the glutamic acid, and so forth. They're able to determine how each copolymer stuck to particular codon sequences associated with a given amino acid, and in that way, they were able to identify a few of the triplet codes associated with given amino acids. And then the last set of experiments, Nuremberg, which we talked about earlier, Marshall Nuremberg, and Philip Leiter came up with RNA triplets. In other words, they made full codons. And each one of those triplets acted functionally just like a codon. And different triplets caused different binding patterns to different transfer RNAs. So it wasn't just that you're dealing with individual fragments of RNA, letting opposite codes bind to each other. These were actual tRNAs that had amino acids hooked onto them. And so in that way, they were able to determine that, say, for example, CCC triplet causes the binding of proline to a transfer RNA. And so to do that, you also had to have ribosomes in the mix. So here is the small subunit and the large subunit. And we'll talk about each of these sites in a minute. Here's the CCC piece of RNA. The transfer RNA carrying proline was the only transfer RNA that could associate with or had a anticodon to CCC, so that proline would fit into this site. And so basically what they did was they looked at the ribosomes that had the CCC incorporated with an amino acid containing transfer RNA. And this was the last piece of evidence that was necessary to identify a given codon to a given amino acid. So then that makes us start to wonder about the structure and function now of transfer RNAs, because we know that transfer RNAs have the anticodons associated with them. So you have your transfer tRNA, and it has an anticodon that binds to the codon in messenger RNA. So there are some structural features of transfer RNA that allow it to do its job. One of it is the amino acid associated with one end, and the other end is an open end. Don't worry about that. There is also an amino acetyl tRNA synthetase that charges each given transfer RNA with the appropriate amino acid. We'll talk about that in a minute. And then those famous Wobble rules, the Wobble, that last amino acid in a codon. What are the Wobble rules associated with the transfer RNA? So what was interesting was all of these experiments were being done, and they all played into Watson and Crick's idea of how DNA is the key genetic information in all organisms that stores the meaningful information to build an organism, right? Francis Crick himself came up with the idea of what's called the adapter hypothesis. And so there was an adapter molecule that connects between the codon and the amino acid. All right, so you had the codon and then the amino acid, and that turned out to be the transfer RNA. So transfer RNAs recognize the 3-base codon in messenger RNA through its anticodon interaction. Pardon me. So this is one of the ways that transfer RNAs are drawn out. So you have the phenylalanine in this case, and in this case you have proline. So tRNA, phe, tRNA pro identifies the type of transfer RNA. But down here at the base, at this curl in the tRNA, is what we call the phenylalanine anticodon. So if the codon for phenylalanine is UUC, the transfer RNA is going to have exactly the opposite sequence so that they can stick together and form hydrogen bonds, okay? And in fact, that's what these little dots are. It's just I'm extending that little bond here. So that U binds to A, U binds to A, and G binds to C, 5' to 3'. Over here, the proline codon, CCG, is associated with, or is opposite of, the proline anticodon on the proline tRNA, which is GGC. Who reads all of this? The transfer RNA and the messenger RNA. That's going to be the ribosome. We'll get to that in a minute. Let's cover a little bit more of the structural features of a transfer RNA. Transfer RNAs form within themselves a secondary structure. So you have the long string primary structure of the transfer RNA, which is just going to be nucleotides. The secondary structure are going to be 3 stem loop structures, so it looks kind of like a clover leaf, which we'll look at. Excuse me. And then a couple of what we call variable regions. The acceptor stem has a 3' single-stranded region, which we'll look at in a minute, that has a special job. And this 3-dimensional or tertiary structure allows folding so that the transfer RNA can be accepted into the ribosome appropriately, kind of like a key. Most of the normal nucleotides are A, U, G, C, as in all RNAs. However, the transfer RNA has a few modified nucleotides, and they're typically going to be uracils, pseudo-uracil, things like that. So you have more than 80 individual sequences to make up transfer RNAs associated with each different flavor of amino acid. So here is our secondary structure. So this is a stem and this is a loop. The 3' end is the acceptor stem, and the acceptor stem is where you're going to see the bond between the transfer RNA and the amino acid, okay? When the amino acid is added onto the 3' end, that's called tRNA charging, okay? I'm writing with my mouse again because I still don't have batteries. So tRNA, love it, charging happens when an enzyme brings the appropriate amino acid to the transfer RNA. And guess what? That enzyme identifies it by looking at the anticodon. Amazing, huh? Over here, the modified bases are the I is iodine, ML is methyl-linosyl, and so forth. Don't worry about that so much. You just need to know that some of these exist inside of the transfer RNA, but you'll never see that in messenger RNA. So charging, that step of adding the amino acid onto the transfer RNA happens, like I said, with transfer RNA synthetases, aminoacetyl transfer RNA synthetases. So there's a 2-step reaction that allows the right amino acid to be bound to the right transfer RNA using, you got it, ATP. Got to have energy for that. So the selection of the right amino acid has to be extremely accurate. Otherwise, you're going to have a mutation introduced because the wrong transfer RNA is carrying the wrong amino acid. Fortunately, the error rate for inappropriate charging is less than 1 in every 100,000 transfer RNAs. That's pretty impressive. So pretty much most of our transfer RNAs are carrying around the appropriate amino acid. So here are the steps. You have a given amino acid. Here is the aminoacetyl tRNA synthetase, the enzyme. This one just happens to take this one specific amino acid, the yellow one, okay? It binds with some energy, with some ATP, which gives a phosphate to the given amino acid. Then the ADP is ejected out. Now you have an amino acid carrying a phosphate. When the AMP is kicked out, the appropriate transfer RNA fits into the space because you have a matching of the codon and the anticodon right here. When that happens, the transfer RNA, the phosphate is cleaved, and actually the phosphate leaves with the AMP, and then you have the charged amino acid. So that leads now to our transfer RNAs and this Wobble rule. I've mentioned this Wobble. So basically, the codon-anticodon recognition process is really fast, okay? And this whole process was proposed by Francis Crick because Francis Crick was a really smart guy. But that third position, okay, the AUGC rule binds real quick during the first initial mRNA and tRNA recognition. That third position actually can go through some mismatching. In other words, the ribosome is kind of paying attention to, for the most part, the first 2 amino acids, because this has to happen really quick. And so by the time the third hydrogen bond is formed between the third amino acid, and not amino acid, the third nucleotide in the codon-anticodon pair, sometimes a mismatch occurs. And so we have what are called isoacceptor tRNAs. Basically what that means is that the same codon happens on the first two, but the last one can have wobble. So here is AU, but the third position can be out of the ordinary, and the ribosome will let that slip through. And so this gives you a calculation. Basically, it shows, all right, this is just not working. Basically, this shows you what the possibilities are for the third base. Okay, got rid of that. So now, what about the ribosome? The ribosome is super, super important. The ribosome has definitely some structural, features. The ribosome is found always in the cytoplasm, all right? So you can find ribosomal subunits in the nucleus, but they are not functional there. They're only functional in the cytoplasm. And eukaryotic cells have two types of ribosomes, all right? We have mitochondria. We have some found in the cytoplasm. There can also be some found in organelles, mitochondria, and chloroplasts. Those separate ribosomes typically are only found in plants because, yes, you have the chloroplastic. But in mitochondria, you'll find that in all eukaryotes, as well as the cytoplasmic ribosomes. So we say two types. I kind of think of it more as three. This would be number one, mitochondrial, which is 2, chloroplastic, which is 3. Wow, that looks really bad. Now, why do they say 2 types? Well, because mitochondria and chloroplasts are believed to be leftovers, or they're believed to have been brought into eukaryotic cells through an endosymbiotic event. Mitochondria are probably leftovers from purple sulfur bacteria, and chloroplasts from cyanobacteria. If you haven't taken Bio-01, we talk about that in there, and we did talk about it earlier in this class as well when we talked about extrachromosomal DNA. So in addition to having extrachromosomal DNA, they have extracellular ribosomes. So each ribosome, like I mentioned on the very first slide, has a large subunit and a small subunit. And in addition to that, there are proteins and rRNAs associated with the formation of, the formation of each subunit in the nucleolus. So the nucleolus is the really dark spot in the middle of the nucleus, and that's where all the ribosomes are made. So here's the small subunit, here's the large subunit, and then assembled together, this is what they look like. We're not going to worry about sedimentation coefficients and how this was worked out. You can learn about that in the next genetics class that you take. Now, ribosomes can occur as individual ribosomes, or, they can occur along a length of messenger RNA to form what we call a polyribosome or a polysome. Now, for a long time, it was thought that polyribosomes occurred only in bacteria, but polyribosomes have also been seen in eukaryotic cells as well. So each little blue spot is a small and a large subunit of the ribosome glued together over the messenger RNA, which is the red line, and then each green line is the forming protein or polypeptide. So you can see that right here is the beginning of the translation region, and as we go farther along, the protein gets longer and longer. So the functional sites within a ribosome are the peptidyl site, the P site, the aminoacetyl site, the A site, and then the E site, or the exiting site. In the past, each site was considered independent of one another. These days, the exit site, there are some labs arguing that the exit site actually doesn't exist. But for the purposes of this class, we are going to stick to A, P, and E.